It's a nice picture of one of the blood vessels, as you can see here, in sagittal section, kind of running diagonal on the board. And we have some red blood cells here, 2 of them running in single file. And this is a beautiful image of the lining of this blood vessel. This is a capillary. This is a non-fenestrated, or what they call continuous capillary, with the most common capillary you find in mammalian bodies and many vertebrate bodies. These flat cells, this simple squamous epithelium that lines the cardiovascular system is called endothelium, is the name given to it. So endothelium is the lining, this simple squamous epithelium that lines the entire cardiovascular system, including the atria and the ventricles. And it is going to provide the barrier, if you will, between circulation, between the blood that's circulating and the interstitial fluid. We'll talk a little bit about blood vessels, a little bit about the endothelium and blood cells in the next part of lecture, which will be on the 3rd exam. So introduction here. All cells have to move materials, right? And we know that. We've talked about membranes and how they move things from the extracellular environment to the intracellular environment. As organisms became multicellular and went through the evolution of multicellularity and whatnot in size, cells started to move further and further away from the surface of these multicellular organisms, which provides a problem. The problem is that if you're too far away from the environment, how are you going to get the nutrients, oxygen, and get rid of the waste products that you need? So as cells, as organisms evolved and became multicellular, several systems had to evolve at the same time in order for organisms to get larger. And one of those is the respiratory system, which moves gases, right? And the other is the cardiovascular system, and they kind of evolved together. The cardiovascular system provides a lifeline, if you will, to all of the cells of the vertebrate body. So there are 60,000 kilometers of blood vessels that run through the average human body. Think about that, 60,000 kilometers. No cell in the mammal is typically more than 2 to 3 cells away from the smallest blood vessels in the body, called capillaries. So the cardiovascular system is really important. And the main reason for that is because most cells in your body are anchored in place. They can't move. They can't get up and just leave if the environment falls apart. So you need a lifeline. You need a system of tubes that are circulating fluid that can deliver you the oxygen and nutrients you need and take away the waste products that you're producing. And so we know about a lot of the transport mechanisms at the cellular level: exocytosis, endocytosis, transport by proteins like facilitated diffusion or active and secondary active transport. All of these things happen at the cellular level of the cardiovascular system also, but we're going to add a mechanism here that eventually we're going to call bulk flow. All right. And bulk flow is basically the movement of materials at a higher rate or a more efficient rate than diffusion alone. And it's going to be provided, bulk flow is going to be provided by a pump we call the heart. The heart is going to squeeze on blood, and that's going to produce hydrostatic pressure. We call that hydrostatic pressure blood pressure. And if we produce a high enough blood pressure, that's what forces fluid through the cardiovascular system. As a vertebrate, we're going to talk mostly about vertebrates. We have a closed circulatory system, which means all the blood vessels are closed from the rest of the body. Therefore, we can maintain high pressure in that system because it's not an open system where the pressure would dissipate. The pressure is going to dissipate slowly over time because of friction, but it doesn't dissipate to zero, if you will. So fluids are going to be under high pressure and moving at a high flow rate, if you will. So when we get to the capillaries, we'll talk about diffusion. Diffusion still is one of the most common ways that we move things, even with the cardiovascular system. But as animals got larger, we had to divide, right, the cytoplasm up into smaller units so that we maintain our high surface to volume area, and that's where multicellularity comes in. And as animals become more and more and more cells, we got to evolution figured out a way, or provided a way, if you will, to deliver materials to the deepest cells of the body. So large multicellular organisms can't rely on diffusion alone, and that's where the cardiovascular system comes in. So the cardiovascular system is basically made up of the 3 elements here, right? So looks like our 3rd element got covered here. So for whatever reason. So these are the 3 elements of the vertebrate cardiovascular system. So we have a heart, which is our pump, right? That is going to help provide the pressure we need to cause flow, all right, or push blood through the blood vessels. We have blood vessels, and there are veins and arteries and capillaries, and we'll define what those are in a minute. And then we have basically two loops, if you will, within vertebrates. We have a loop of blood vessels that's between the heart and the lungs. That's called pulmonary circulation. And then we have a loop of blood vessels that leaves the heart and supplies the rest of the body, and we call that systemic circulation. And then obviously what we're pushing through these blood vessels is blood, right? And blood is a composite structure. It's a specialized connective tissue, and it's a suspension. It's made up of a liquid portion we call plasma, and then formed elements that we call blood cells or pieces of blood cells. And again, we'll go through those in the second part of the cardiovascular lecture. So this is kind of the setup here, if you will, of the vertebrate circulation. So we got our centralized heart. This is a mammal. We have 4 chambers. Basically, the heart in a mammal is 2 separate pumps, if you will. It's a right pump and a left pump. And we have a set of blood vessels that attach the heart to the lungs. In this instance, since we're tetrapods, right, as mammals, and breathe air. And that's called pulmonary circulation. And then we have a loop of blood vessels that connect the heart to the rest of the body cells, and that's called the systemic circulation. So we're going to talk about the heart. We'll use the mammal heart mostly as our model, but I'll show you a couple of other hearts. And then we're going to talk about how the heart works, because it works similarly in all vertebrates. All right, so here are some of the major functions of the cardiovascular system. Some of these you've already know, probably most of them you've heard of, right? So the circulatory system transports gases between the 2 types of respiration, or the 2 levels or sites of respiration, if you will. They're called external respiration and internal respiration. External respiration happens between the blood and the environment. So it's going to happen at the lungs or gills, or even the skin in amphibians. Internal respiration happens between the tissues and the blood. So at the capillaries and the tissue cells of the body. The cardiovascular system adjusts and maintains things like pressure and flow rate. Think about the different pressures that an animal, like a diving animal, for example, like a sperm whale, which will dive thousands of feet to hunt squid. All that water on top of their body column really changes blood pressure. Or if you're a giraffe with your head up and you put your head down to drink and you bring it back up really quickly again. Think of the pressure changes that a giraffe has to deal with. The blood transports excess heat from the body, brings it to the surface, and we sweat, right? So we can evaporate off heat. Cold-blooded animals also bask and send blood to the skin to bring in heat, right? To gather the sun's energy, the infrared energy of the sun. Transports nutrient molecules, glucose, amino acids, fatty acids, anything that we need. Vitamins are going to travel from the digestive tract to all of the body cells. Transports hormones, which are communication molecules. We're going to cover that chapter in the endocrine system. Transports cells' waste products to the kidneys for disposal or to the liver for recycling. The liver and kidneys play vital roles in the recycling and removal of waste products, or the detoxifying of toxins that might get in the body. The blood also carries a series of chemical cells and weapons, if you will. White blood cells, which can themselves kill invaders, or they can produce weapons like antibodies that can destroy invaders. And so the blood protects the body, which is nice. So, flowing through those 60,000 kilometers of blood vessels and out to all of the body tissues are white blood cells, which help with innate immunity and also specific immunity. It also participates majorly in pH, as we have kind of already discussed, with that bicarbonate carbonic acid system. And bicarbonate is the main blood buffer, and therefore the main buffer of the interstitial fluid also. So those are kind of some of the major functions, if you will, of the cardiovascular system. Central plan of the cardiovascular center includes these 4 components. So we're going to really focus today on talking about the heart for most of the rest of this lecture. But I'll introduce you really quickly to the arterial system, the capillary system, and the venous system here. And we got probably the simplest circulatory system in vertebrates, which is the fish. And notice they have really just one loop or one circuit. You see a pulmonary and a systemic circuit only in tetrapods, organisms that breathe air. And that also includes the lungfish. So some fish have both loops if they have at least a 3-chambered heart. And the lungfish do have that 3-chambered heart because the pulmonary system is going to supply their two lungs, which are swim bladders. And the rest of fish, they have a two-chambered heart that is linked to two major capillary beds, the capillary beds of the gills and the capillary beds of the body. And so the capillaries are the smallest blood vessels in the body. They are small enough to only allow red blood cells to move through them in single file. They also have the thinnest walls, and that are the only place in circulation where we exchange materials between the blood and the tissues. And they also connect the arteries and the veins. So the easiest way to define arteries and veins, arteries are blood vessels that take blood away from the heart. That's what an artery does. A vein brings blood back to the heart. Most arteries carry oxygenated blood, and most veins carry deoxygenated blood. When we get to the pulmonary system, however, the veins carry oxygenated blood, and the arteries carry deoxygenated blood. That's why we use toward or away from the heart. Capillaries connect the two. So arteries typically drain into a capillary bed, and then that capillary bed is going to drain into a vein, and the vein will then return the blood back to the heart. The arterial system includes the largest blood vessel in the body, which is the aorta. It also has the thickest wall. Arteries hold their shape and have either a lot of elastic connective tissue in their wall or a lot of smooth muscle. Veins are wider and thinner, tend to not hold their shape, and the largest veins in the body are the vena cavas, the superior above and the inferior below the heart, that bring all of the blood back to the heart. Again, capillaries, the smallest blood vessels. The smallest arteries are going to be called arterioles, which I've already defined for you before when we were in the kidney. And then arterioles drain into caps, or capillaries. Capillaries drain into the smallest veins, which are venules. So these three blood vessels are called microcirculation. They include the smallest blood vessels of the cardiovascular system. The most common blood vessel, the capillaries. Capillaries make up most of that 60,000 kilometers of blood vessel. So central circulation revolves around a heart. And basically, if we bring blood back to the heart, we're going to bring it to the atrium or the atria, depending on whether we have one or two of them. And it's the vena cavas that return blood to the right side of the heart. The heart will either go right out to the body if it's a fish, or to go into pulmonary circulation to the lungs and back to the left side of the heart. The left side of the heart, blood will be pumped out of to the largest artery, which is called the aorta. Then the aorta will deliver blood, oxygenated blood, to the body. So that's a little bit about the central plan, a little bit about the blood vessels. Again, we'll spend a little more time talking about the histology of the blood vessels and how veins and arteries differ as we do the second lecture, which will be part of the third exam. All right, so again, just showing you there are a couple different figures here. So we got a heart, our central propulsive organ. We have veins and arteries and capillary beds, right? We do have some fish and then invertebrates that have multiple hearts, not just one. Or we can have an axillary heart, a heart that, or an accessory heart, a heart that helps somewhere in the body in some other organisms that will actually help pump, let's say, from a tail or from a long limb. In earthworms, we have axillary hearts that run along the head of the worm that connect the two aortas together. Again, you can have many pumps or one single central pump. There's no structural support in vertebrates. There's no turgor pressure, no hydrostatic pressure that gives our body its kind of support. Our body is supported by our skeleton. Our vessels are under very high pressure, somewhere around 100 to 120 millimeters of mercury, which is a ton of pressure, passes through our circulation. We also have low volume of fluid. Only about 5% of our body fluid is actually plasma. High flow rate, very efficient as far as circulation goes. The heart's very efficient at moving blood through those 60,000 kilometers of blood vessels. Capillaries, we have what's called ultrafiltration. So the capillary wall is either continuous or it has small holes in it, like we saw in the kidney, and can control what passes through. If it's a continuous capillary bed, that is, materials would have to pass transcellularly through the cells using specific proteins or specific channels or endocytosis. If I'm a fenestrated capillary, then it's going to be based on size of the pores, right? And how much of the plasma or what can leave based on size. So we call that ultrafiltration because they're very small pores. So here are a couple different plans that show the vertebrate circulatory systems. If you were taking like comparative anatomy with me, we'd spend some time talking about the differences here. But in reality, as you can see, what happens here is we move from an aquatic environment to a terrestrial environment as we include another loop, right? And that loop is going to include our respiratory organ for air, which is either skin, lungs, in our examples of tetrapods. And this pulmonary circulatory part of our circulation is involving just the respiratory organ. It connects the respiratory organ to the heart. And then the other loop is the systemic loop, which connects the rest of the body to the heart. You can see we start with a 2-chambered heart, go to a 3, and then 4-chambered heart. And the big difference between the 4-chambered hearts in reptiles, or the 3-chambered and 4-chambered heart in reptiles versus mammals and birds, is that the right and left side, our two pumps, are completely separated in mammals and birds by a septum, so that the blood on the right side can't mix with the blood on the left side. The hearts are very efficient in all of these circulatory patterns as far as producing the pressure to circulate blood. So the primitive form here is the 2-chambered heart in the 1 loop. The intermediate form is the 3-chambered heart with 2 loops. Also in the intermediates, with frogs, we have not only the lungs, but the skin is a major respiratory organ. And then as you move to reptiles and mammals, you're going to move to a 3-chambered heart that functionally is 4-chambered based on how the flaps work. And then with mammals and birds, you have a 4-chambered heart functionally and structurally. The other major difference between veins and arteries are valves. Veins have valves, as does the heart, and valves, their main function is to keep blood moving in one direction. And that's what the valves do, prevent backflow, if you will, both in the veins and in the heart itself, in the chambers of the heart. So we got valves there. Unidirectional flow. Valves allow for unidirectional flow. They prevent backflow. Blood moves in one direction. Okay, again, 2 systems, pulmonary, systemic. You can see within, for example, mammals, systemic has much higher pressure in it. And the reason for that is because you got to push blood through a lot more blood vessels. The systemic circuit is where most of that 60,000 kilometers are. If you think about it, the heart and lungs are right next to each other, right? So there's not a lot of length, if you will, connecting the heart to the lungs. There's a ton of length that connects the rest of the body to the heart. And so most of that 60,000 kilometers of blood vessels are found in the systemic circuit. You need a lot more pressure to force blood through all of those blood vessels. So those are your 2 circuits, your 2 loops. Okay, let's start looking at the heart, right? So this is these are my pictures of mammalian hearts. And this is basically if I took a heart out of the body and cut the top of the heart off, picked up the top and looked down. So you're looking down into the heart, and I've cut the atria off of the heart. And if I just back up one slide here, whoops, the wrong way. There are 2 types of chambers in the heart. If you don't know that already, they're the atria and the ventricles. The atria are on top. The ventricles are caudally located. The atria are smaller. The ventricles are larger. The ventricles are also more muscular. That's because they push blood farther. The left ventricle is the thickest portion of the heart because it has to force blood throughout systemic circulation. So if we remove the atrium and looking down, we can see our 2 ventricles. Got the right ventricle over here, the left ventricle on this side. I know this is the left ventricle because the wall's much thicker, as you can see, compared to over here, because you have to force blood to through more blood vessels. This is the septum here, completely separating the right from the left ventricle. So you know this is a mammalian heart. There's no way blood on the right side can mix with blood on the left. This is a sagittal section through a heart, gross anatomy-wise up here. This is the atrium. Down here is the ventricle. Here are the 2 flaps. This is the bicuspid valve. This is the left side of the heart. Notice these cords that look like tethered rubber bands that are running from the edge of the valve and attached to the wall of the ventricle. These are called chordae tendineae. And the chordae tendineae, what they do is when the ventricle contracts and pushes blood up, the valve is going to close, right? Like this. And what the chordae do is they pull as they're attached to muscle too, as the ventricle contracts, they pull down. So when that blood hits that valve, it doesn't pop back up into the atria. So what the chordae tendineae do is prevent the flaps from prolapsing. All right. And then blood would backflow right into the atria, which we don't want. We want blood to swirl under, the valves and then go up into the aorta. So that's what the chordae tendineae do, is they prevent the valves from popping into the atria, which is called prolapse, and allowing blood to flow back into the atria. Again, the valves allow for one-way flow. Now, if we go down and we take a look at a piece of heart muscle under a microscope, this is what cardiac muscle looks like on the bottom image here. This is skeletal muscle on the top image. You can see cardiac muscle is highly branched. It is a striated muscle. It has stripes in it, just like skeletal muscle does. And so the two types of striated muscle in the body, as you already know from histology, when we had our little histology section, are skeletal muscle and cardiac muscle. Smooth muscle lacks the striations, right? Hence its name, smooth. Smooth muscle also functions completely different than cardiac and skeletal do because of its difference in morphology. Cardiac muscle and skeletal muscle both shorten in the same way, the same mechanism. They're organized the same way as far as the inside of the cells and the sliding filament theory, which we'll learn when we get to muscle tissue. But the shortening of these muscles is what produces force, right? A cardiac muscle squeezes or shortens and squeezes on the blood. If you can imagine a balloon with water in it, right? If we squeeze the walls of the balloon, leaving the top untied, the water squirts out, right? The top of the balloon, the same thing with the heart. The walls squeeze on the blood that fills the heart and forces it into a blood vessel. So when we look at how cardiac muscle contracts or shortens, we're going to do it briefly here and then focus more on the electricity that the heart produces and how electricity spreads. We will talk a little bit about contraction. The one thing that you'll notice, three major differences really that you see in cardiac muscle off of this picture is that the cells are branched. The cardiac muscle cells are branched. You can see here's a very good example where my laser pointer is, where these cells are cylindrical and unbranched, the skeletal muscle. They're also intercalated discs or dark lines, as you can see, I'm pointing out here, that separate one cell from the other. Skeletal muscle is much longer and larger than cardiac muscle. So you got shorter cells. And the nuclei, here's the nucleus right here, I'm circling it, is essentially located in the cardiac muscle, right in the middle of the cytoplasm, whereas the nuclei in skeletal muscle are located on the periphery. So those are a couple major differences. The other thing is each cell here of cardiac muscle only has one nucleus, maybe two sometimes, but typically in a mammal, just one nucleus, whereas skeletal muscle can have up to 30 or more nuclei. So it's multinucleated, one of the few multinucleated tissues in the mammal body. So those are some of the differences. There's also some major differences with the membrane channels and proteins that are found in cardiac muscle and the way the muscle behaves electrically. Contraction-wise or shortening-wise, cardiac muscle it has the same mechanism as skeletal muscle. All right, so I kind of talked about a lot of this already in the last slide. I'll give you some pictures here. So we got highly branched cells. They form a network that is hard to pull apart, and that's important as the heart stretches or fills. We don't want the cells to be separated. The cells are also glued together or spot welded together at the intercalated discs. So these intercalated discs, what they are a combination of two major junctions, desmosomes and gap junctions. All right, so the desmosomes keep the cells from being separated, and the gap junctions connect the cytoplasms. And that's really important because we need spontaneous coordinated control of each of the networks of the heart. There are two major networks of muscle that make up the mammalian heart: the atrial network, the network of muscle on the top, and then the ventricular network on the bottom. There's no direct connection between atrial cardiac contractile cells and ventricular cardiac contractile cells. So we're going to have to talk about how, with pacemakers and specialized cells, we connect the atrial network to the ventricular network. The biggest thing with contraction of the heart is we need coordinated contraction. We need all the atrial cells to contract at the same time, all the ventricular cells to contract at the same time, so that we get equal and smooth force or transition of force in both networks. These cells have enlarged endoplasmic reticulum. They also have some deep cell membrane invaginations or tubes that dive down deep into the cytoplasm that are called T-tubules, as skeletal muscle also has those. And Cardiac muscle, there are fewer T-tubules, but they're much more branched. They're highly branched and interconnected, and they form a network of canals that connect the cell membrane to the deepest part of the cytoplasm. Probably the neatest thing about cardiac muscle is its ability to spontaneously contract. And so it contracts on its own. You can take a heart out of the body and it will contract all by itself. We will do a lab that is virtual that I will show you here in the next couple weeks. That is a frog lab working with the heart and how the heart reacts to certain hormones or ligands, communication molecules. But I also take the heart out at the end and place it on the table, and it contracts all by itself and continues to contract. There's nothing in the body that tells the heart to contract. Now, your nervous system can take over and speed the heart up or slow it down, but the heart controls its own beat, its own rhythm, if you will. And it does that intrinsically. There are specialized cells inside of the heart called pacemakers that actually produce the electrical current that causes contraction. And so there are certain cells within, or really all cardiac muscle cells have the ability to spontaneously contract. And they will produce their own electricity, which produces contraction. The fastest spontaneously depolarizing or spontaneously electrical producing cells in the heart are called pacemaker cells. And these pacemaker cells are what cause the entire heart to contract, because the fastest one to produce this electricity will then allow it to spread through the gap junctions. And then all of the networks, both the atrial and ventricular networks, will end up contracting. The gap junctions coordinate the contraction so that your two networks, all of the cells of a network, contract at the same time. This electrical current that is produced spontaneously are called action potentials. And action potentials are complete reversals of membrane potential. We already know what membrane potential is. We'll go over it really quickly again. But we know all cells have negative membrane potential. Cardiac muscle cells have some of the most negative membrane potentials at around negative 90 millivolts. And action potential is a complete reversal of that. So it goes from negative 90 to positive 30. So you reverse the membrane potential completely, and that positive charge spreads across the surface of the heart. So it's not only a change in membrane potential, but it's also the spread of that change in membrane potential across the entire surface of the heart and all of the cells, which is called electrical current or electricity. All electricity is the flow of a charged particle. The mammalian heart is made up of two major tissue types, cardiac muscle and then connective tissue. All right, the connective tissue anchors the heart in place. There's also some fat around the heart, also forms the valves and part of the kind of what we call the connective tissue skeleton, if you will, of the heart itself, giving the heart its shape. All right, so cardiac muscle, let's focus. There's going to be kind of two types of cells, two types of what we're going to call muscle cells, all right? We're not going to focus on the connective tissue as much here. We're going to focus more on the functional cells of the heart. So there are contractile cells, and those cells are the ones that shorten and produce force, right? And they squeeze on the blood. And then there are autorhythmic cells or pacemaker cells, and they basically produce the electricity spontaneously and conduct that electricity to the right places. And that's really their main job. They do not shorten because they do not have what are called organized sarcomeres. They don't have the striations, the light and dark striations that you see. They're typically smaller and fewer in number and less branched. And they are going to form networks that are going to form the pacemakers and then the conducting system of the heart. So this is a picture, as you've already seen, histologically of good old cardiac muscle. So you can see the centrally located nuclei, branched, striated, right? These are contractile cells. And then the white, these are stained differently. So these white lines are the intercalated discs that cement the cells together, spot weld the cells together, and connect them through gap junctions. So as I mentioned before, this is an electron micrograph that's lightly stained. And you can see this is an individual cell. Here is the cell membrane here. There are basically long fibers that are found inside of both skeletal and cardiac muscle. These long fibers, or what are called fibrils, are called myofibrils. They're long rods of proteins that are made up of actin and myosin and cementing proteins. These long rods are broken down into individual subunits or squares, rectangles really that are called sarcomeres. And it's the sarcomeres that actually shorten. And so if I take a long fibril, all right, or I fill the cytoplasm with long fibrils of all of the same length, and I shorten all of the sarcomeres of those myofibrils, all the myofibril rods are going to shorten. And if they're connected to the cell membrane, which they are, they're going to shorten the entire muscle cell. And that's what myofibrils do. So the inside or cytoplasm is packed full of these long rods made up of actin and myosin. And the actin and myosin shorten, which shortens the entire rod. And these rods will then shorten the entire cell. What causes them to shorten is electrical current and the release of calcium. Calcium is a major second messenger, and calcium is released in response to electricity. The calcium comes in through calcium channels, you can imagine, that are voltage gated. The calcium flows in. The myofibrils, these rods, shorten inside the cytoplasm so that the whole cardiomyocyte, the contractile cardio heart myocyte muscle cell, shortens. What delivers the electrical current to these myofibrils, these deep rods, is a tubular system called T-tubules. And you can see they're highly branched. I colored them green here so you can see them. They originate up in the cell membrane, and they basically dive down, and they're filled with interstitial fluid. So they're long straws of cell membrane. And these straws have certain proteins embedded in their membranes, which are voltage gated. Some of them are calcium channels. Some of them are maybe potassium channels, maybe sodium channels, depending on whether we're talking about skeletal or cardiac muscle. So this is a low power image, a 4X image of the heart. This is the surface of the lumen, the deepest part. This is the lumen of the ventricle. Here is the endothelium. You remember the endothelium? What's the endothelium? The endothelium is the simple squamous epithelium, right, that lines the entire cardiovascular system. So the endocardium includes the endothelium and its basement membrane, and then some connective tissue that cements the endothelium to the underlying wall of the heart, which we called the cardiomyocytes, the contractile cells. Notice in this picture, you have two types of cells. You have these lighter staining cells on top. They're almost the same color as the endocardium. These are the Purkinje fibers. These are part of the pacemaker cells. Those modified cardiac cells, they conduct electricity. Then you got these darker bundles of cells down here, which are the contractile myocardiocytes. They contract. The majority of the wall of the heart is cardiomyocytes that contract or shorten to squeeze on the blood. And these cells are in cross-section, as you can see here, whereas the Purkinje fibers are in sagittal section. So this shows you an SEM of cardiac muscle. The contractile cardiomyocytes. This is a little segment of cytoplasm. So if I zoom in on one of the inside of those cells in the cytoplasm, you see two of these rods in this picture. There's a rod up here. Here's one rod running across the screen. Here's another rod. You can see the rod, if this is one rod here, is broken down into functional units that are rectangles, right? And each rod has the same number of these functional units, and they stack up real nicely like coins. These are called sarcomeres. These smaller striations that you see inside are the actin and myosin. So this whole rod, you can imagine the sarcomere shortens, these two sarcomeres shorten, right? In response to electricity, the whole rod shortens. Same here, up here on top, the whole rod, if these two rods shorten, they got to pull the whole cell in, right? Because the rods are attached to the cell membrane. So that's how shortening works, all right, and which is not something I'm really focusing on. I just want you to know that there are myofibrils that make up, right? These are myofibrils that make up the muscle fiber here. So this fiber here is a myofibril. There are actually two of them. One, two, right? Two muscle myofibrils. These are mitochondria here. And then these clear areas here are parts of the T-tubules, right, that are delivering what we're going to call the action potential or electricity directly here to the sarcomeres. And what they're delivering the electricity to is going to be the endoplasmic reticulum, which is the other major component here that we need to talk about. The ER in skeletal muscle and cardiac muscle sequesters or stores calcium. And so these T-tubules are going to come down right next to the ER and stimulate them to release calcium, because the ER here has calcium voltage gated channels that open in response to a change in membrane potential. Calcium is going to flood the cytoplasm and it's going to flood the sarcomeres, and the calcium is going to cause contraction. Right? So that's the signal. The electricity causes that second messenger calcium to flood the cytoplasm, and that causes these myofibrils to shorten and the sarcomeres to shorten, and the whole muscle cell will shorten, squeezing on the blood and forcing it through the arteries and veins. The mitochondria make the ATP that drive this, right? You need ATP in order to shorten the sarcomeres. This shows you one cell on the bottom and one cell on the top, and this is an intercellular canal between two cardiomyocytes, and these pictures here, the darkenings, are the desmosomes with gap junctions. This is an intercalated disc that cements these two cells together and connects them via gap junctions. So that shows you that junctional complex we call an intercalated disc. That is a combination of gap junctions and desmosomes. Those dark protein accumulations should look familiar. It's what we've talked about earlier in the course, which are structures we call desmosomes. Again, on the bottom here, we have actin and myosin labeled for you. That's what the stripes are. The actin and myosin, remember, make up the myofibril, which is divided into sarcomeres. And that we have one myofibril kind of running up here, another myofibril running up over here, and then one running down here, right? And notice they connect into, right, the cell membrane there, and that's why they pull or shorten the whole cell when those sarcomeres shorten. Okay, again, we're going to talk more about the comparison between skeletal smooth and cardiac muscle. Right now, we've kind of talked about skeletal and cardiac a little bit. They both appear striated because the inside of the cytoplasm is packed with rods that have light and dark bands, which are called myofibrils. Those myofibrils are broken up into sarcomeres. The sarcomeres are what shorten all of the fibers we call myofibrils, which shortens the entire cell. So they're arranged in sarcomeres. Notice smooth muscle has no sarcomeres. Location, obviously skeletal muscle is associated with the skeleton, right, and moves bones. Cardiac muscle is associated with the heart. We have multinucleated large cylindrical unbranched fibers for skeletal muscle. We have uninucleated shorter branched fibers for cardiac muscle. Actin and myosin are the major proteins here. Also notice that, and they make up sarcomeres, which make up the entire myofibrils, right? Also, we have two proteins here called troponin and tropomyosin. Right? And cardiac and skeletal muscle, those are the regulatory proteins. They bind calcium. So when calcium is available, it binds to troponin and tropomyosin, allowing the sarcomeres to shorten. The calcium, again, is released because of electricity or an action potential that's produced, causing the release of calcium from the endoplasmic reticulum. Control, calcium and troponin, right? Troponin and tropomyosin is what causes contraction. Calcium and troponin here, they are both electrically stimulated, right? This particular, notice in cardiac muscle, the electricity spreads through gap junctions, which skeletal muscle does not have. So notice that difference. We have gap junctions here that control all of the cells of a network. There's an atrial network and a ventricular network, right? An atrial network and a ventricular network. If we come down here to initiation of contraction, requires a neurotransmitter called ACH, which is acetylcholine. That's what ACH stands for. And a motor neuron. So you need nervous control here of skeletal muscle. Notice under cardiac muscle, we got autorhythmic. That is, the cells produce their own electricity. We'll talk about epinephrine versus acetylcholine when we get towards the end of this lecture. We won't make it there today. All right, so let's talk about this electricity. We've really already got all of the background that we need to know, and I'm going to skip over some of this relatively quickly because you already know this background. We've already gone over it. We're going to apply all of that foundation that we talked about in the beginning of the course. So electrical currents are essential for animals. Animals produce a lot of electricity. The electricity that we produce controls how our nervous system works and controls muscle contraction. Really important in the heart. Think about the heart is basically producing the electricity all day long because it has to contract all day long. And it does this 24 hours a day, 7 days a week, from the time that the heart develops all the way till the day that you die. right? And that electricity is what is causing the contraction or rhythm of the heart. So water is important because it can conduct electricity because water has ions in it, right? You add some electrolytes, particularly like sodium, chloride, calcium, potassium, and amino acids and anything that's charged. And now you have a fluid that can actually conduct electricity very efficiently. And so because we are a vat of water and electrolytes, we have the ability to conduct currents, and our cells are able to produce those currents. So when you set up a conducting bath to actually test how muscle works, you will produce basically a tank, if you will, that has ions in it, right? And in this particular tank, we're using potassium chloride, which is a salt. If we add that to our fluid, it's going to break down into cations and anions. If we add 2 electrodes here that are attached to a battery, and we connect the ends of those battery, the positive and negative end, what's called the cathode and the anode, then electrons are going to start flowing along the wires. The electrons are going to flow to one side and attract the potassiums because electrons are negative, right? The other side, where the electrons are being pulled from, is called the anode, and the chlorides go towards that, which is going to end up being the negative side. So you're going to get electrical current flowing here, right? Like this. So you get electrical current as calcium and potassium, as chloride and potassium moves. And so the ions start moving. Not only are electrons moving along the wires, but the ions move. And when ions move, it's electricity too. So it's just not electrons like you learn in physics. It's actually ion movement in the body that is electricity. So in our bodies, we move calcium, sodium, and chloride, and potassium. These major ions that are found in our body fluids are what are going to move across the membrane and flow along the membranes of cells like nerves and muscle that are going to produce electricity that is going to change membrane potential. This you already know. I'm not going to go over this figure again. This shows you membrane potential. You need to be able to understand this before you go any further. Right? So make sure you can go over this, take out a piece of paper, make sure you can write it all out. Remember, the driving force here are cells that have leakage channels, and all cells have more potassium leakage channels than they do sodium. That means potassium leaks out because potassium's high inside the cell and low on the outside. Sodium leaks in because it's high outside the cell and low inside. Sodium potassium pump maintains these differences of sodium concentration and potassium concentration. That leads to a slightly positive extracellular leaflet of the cell membrane and a slightly negative intracellular leaflet of the cell membrane. We have a battery, right? This is called resting membrane potential. It's an electrochemical gradient and a form of potential energy. All cells do this. Nerves and muscles utilize it to produce electricity. I guarantee on the test, I'll ask you what is resting membrane potential and how does cardiac muscle maintain it? And you're going to need to draw this, write it out, explain it to me. Now, what we're going to do is use that membrane potential to produce electricity. And that's what I'm going to teach you next. Okay, so electrical properties here. Because we're separating a positive and a negative environment, right underneath the cell membrane, remember, the extracellular fluid and the cytosol are neutral electrically. It's just we have at the membrane this transient flow of electricity, positive on the outside, negative on the inside because of the difference in potassium and sodium leakage channel, and because of the electrogenic properties, 3 sodiums out, every 2 potassiums in. This is called a battery. So this is a biological battery. Cells are batteries. They're biological batteries. This current that has positive on the outside and this negative that you read, this negative membrane potential, is called capacitor current. Right? Your membrane is a capacitor. It stores charge. It's storing positive charge on one side and negative on the other side. That's what a battery is, right? It's a temporary capacitor. Connect the ends of a battery and then electrons flow. If you connect the extracellular and intracellular environments, ions flow, right? And that's electricity. Electricity, simply the flow of a charged particle. In a biological system, it's an ion that's moving. So we work in millivolts. These are really small batteries. This electrochemical potential, because of resting membrane potential, we're going to utilize and form kinetic energy. We're basically going to connect the ends using proteins, right? Using channels, which allow ions to flow. Once ions start flowing, that's kinetic energy. That's electricity. So channels allow for our connection between the positive end, our cathode, and the negative end, our anode, right? And so we get conductance here. Conductance is when ions flow from one side to the next or down the membrane if there are multiple channels. Conductance is hindered by resistance. That is, only a certain number of ions can fit through our circuit here. So notice we got a positive end and a negative end and a resistor. This is a circuit, right? If this is a sodium channel, we open it. Sodium is going to flow into the cell at a certain rate, and that is going to be detectable. It's electrical current. Conductance. You can increase the conductance of a membrane by increasing the number of channels and adding more membrane, right? The more channels and membrane you add, the more permeability you have, the higher the conductance of the membrane. You take the channels away, less conductance. Pretty easy, right? Or the other thing you do is concentrate even more ions on one side versus the other. Add more pumps, and you can get higher conductance because you're going to pump more sodium over, more potassium in, and it'll flow faster. So more channels is typically what you do to get higher conductance, or more membrane and more channels. So this shows you an action potential. So notice we have resting potential here. Remember, resting potential happens all the time, 24 hours a day. The pumps are always working. Leakage channels are always leaking. Cells are at rest are always negative, right? Have a negative membrane potential. Cardiac muscle cells have anywhere, depending on the animal, from frog to mammal, anywhere from a negative 70 to a negative 90 millivolt resting membrane potential. You can see over in this example here, we have a negative 60 resting membrane potential here. When an action potential happens and channels open, allowing positive charge to flow in, what's going to happen is a reversal of your membrane potential, right? What happens here is eventually, notice between resting membrane potential and threshold, you have a stimulus. Something happens. Charge flows in. Some ion is flowing into the cell, which is making the cell more positive. So let's just say, for example, you open a few sodium channels. All right, some sodium channels open in the membrane. Sodium flows in, and you go from negative 60 to negative 40. Negative 40 is threshold. What that means is in the membrane of muscles and nerves, you have certain channels that are voltage-gated channels that have a threshold of negative 40. When they hit negative 40, that threshold, what that means is they change shape. When they change shape, they open, and then a bunch of ions flow in. All right, so if you have a bunch of voltage-gated sodium channels in the membrane that have a threshold of negative 40, when that membrane hits negative 40 from the flow of positive charge, these channels change shape, they open, and then a bunch of sodium flows in, which completely reverses your membrane potential. That's called an action potential. That's an electrical current that you can measure. That is an action potential that happens in skeletal muscles. Notice you get this ascending arm up to 50, then that's when those channels close, and then you descend really quickly because at this point where you get the descending back to negative, right, these are potassium channels that are opening. All right, so sodium channels open and potassium channels open. The sodium channels open first and fastest. They open and close. They close at this point at positive 50. And then the potassium channels finally open and you go back negative. You should understand that go back negative because a bunch of potassium is escaping, right? Because potassium's high in the cell and low outside the cell. And then it takes a second for them to actually close and you actually dip below resting membrane potential, right? So you notice that you actually dip below negative 60, and then eventually you return back to 60 because of the sodium potassium pump and because of leakage. So you're going to have a couple different types of channels that are opening here. So you reverse membrane potential, and then you bring it back to the negative. Right? That negative is caused because of the opening of potassium channels. This is called a depolarization because it's a reversal in negative membrane potential. So you go from negative to positive. Depolarize means to make something more positive. This hyperpolarization below resting membrane potential, hyperpolarization, make it more negative. Hyperpolarization, more negative. Depolarization, more positive. After you get this shock of electricity in a cardiac muscle cell, calcium's released in response to that. Some of these channels are calcium channels. They open in response to this reversal membrane potential. That causes the muscle to contract. So right after the electrical current, once you hit threshold, notice what happens. You go from relaxed heart to contracted heart. See how the ventricles both contract? They're smaller, they're squeezing. Space lessens, they squeeze on the blood, and that forces it out into the blood vessels. So you should be able to define these: threshold, action potential, sodium channels, reversal in membrane potential, and depolarization versus hyperpolarization, right? So versus hyperpolarization. Make sure you have all these concepts down before we move on as you go from slide to slide. So allowing certain ions to move in or out of the cell is going to change membrane potential, right? So we know we have resting membrane potential. And as I mentioned, if you have some channels that open that make it more negative, that's called a hyperpolarization, to make it more negative. If you have open channels that allow ions in that make it less negative, then that's called a depolarization. Depolarization excites muscle and nervous tissue because it pushes it closer to threshold. Hyperpolarizations do not excite cells, right? They push them further away from threshold. So we want to depolarize until we hit threshold. And once you hit threshold, then a bunch of voltage-gated channels open. So that's the difference between depolarization and hyperpolarization. The difference between a depolarization and an action potential is a depolarization can be really small, change membrane potential just a little bit, make it a little bit more positive. An action potential is a complete reversal, right? That's the major difference. Remember, the sodium-potassium pump and leakage are what are maintaining resting membrane potential. So we got resting membrane potential, we got action potentials, right? We got small changes in membrane potential, which either negative hyperpolarizations or small positive changes, depolarizations. Action potentials are also large depolarizations, right? Hopefully, so far, so good. All right, so let's compare skeletal and cardiac muscle as far as when the electricity happens and when muscle contraction happens. Okay, so if you look at these two figures, the one on top is skeletal and the one on the bottom is cardiac. What I want you to notice immediately is that the muscle contraction's in red and the action potential is in blue. And if you look at the two, notice the muscle contraction looks the same, right? It's going to basically be almost a bell curve, right? So you get this slow rise to contraction, peaking of contraction, and then slow relaxation, right? The big difference is the action potential, how fast it is, right? So notice in skeletal muscle, action potentials are really fast. That is, the change in membrane potential that completely reverses membrane potential is up and down. It's really fast, right? It's in milliseconds, about 8 milliseconds is how long the depolarization is. That is what we call an action potential. The action potential on cardiac muscle is much longer and drawn out. It has this really long plateau phase. It's about 300 milliseconds. So once you depolarize, notice you go up really fast, just like in skeletal muscle, and you start to drop, but then you plateau. Something maintains the positive charge for a lot longer time. So notice that the contraction and the action potential look very similar. What this means is you can't stimulate cardiac muscle to contract again during its contractile state. All right. So it's going to completely contract and it's going to completely contract and completely relax before you can stimulate cardiac muscle again. Skeletal muscle, you can contract and contract it harder before it relaxes. You can actually contract it about 90. You can make it contract about 90 times a second. You can make it do this. You don't want your heart to do that. That's called fibrillation. It's called a heart attack. Blood's going nowhere. You want it to contract, squeeze blood, relax. Contract, squeeze blood, relax. You never want the heart doing this, right? All right, that's called fibrillation. You don't want that happening. So the reason it doesn't happen is because the action potential is drawn out. Notice you can keep hitting this with action potentials many times if it only lasts 8 milliseconds during its contractile stage. Here you can't do that because the action potential is done right here when you can stimulate it again and the muscle is completely relaxed at that point. That prevents fibrillation or too many contractions happening in a certain time frame. So cardiac muscle has these very long action potentials, and you might say, well, how and why? It's all about what proteins are found in the membrane. The proteins, the channels these gated channels that are voltage gated channels, right, are much different in what they look like, what amino acids make them up, and how fast they open and how fast they close. Obviously, they open and close much slower in cardiac muscle. And we'll talk about what channels do what. So prolonged contraction, the advantages of this is complete squeezing, produces lots of force, that's sustained force, and then complete relaxation allows the heart to fill all the way back up before it contracts again. That's really important. In skeletal muscle, you want to move really quickly, right? To move around, you might need to contract harder or faster, depending on what's going on. So this prevents summation, or what we call tetanus. So tetanus is when you have lots of contractions happening in a small time frame. We don't want the heart to do tetanus, right? That's called fibrillation. We don't want a heart attack, right? So we prevent summation or tetanus, and we provide this basic rhythm: contraction, relax, contraction, relax, contraction, relax, right? You got a rhythm to that. Skeletal muscle, very short action potential, very short refractory period, is able to undergo tetanus, right? Multiple contractions within a certain time frame. All because of what their channels look like. Pretty cool, right? So one of the neatest things about skeletal, or about cardiac muscle, we're going to focus on cardiac muscle. We'll do skeletal muscle eventually, right? We'll do a whole chapter on skeletal muscle. Is that cardiac muscle can spontaneously depolarize. It can spontaneously produce its own electricity. It's myogenic in the sense that it produces its own or generates its own electricity in its own contraction. So cardiac muscle, like smooth muscle, has the ability to do this, to spontaneously depolarize. When it spontaneously depolarizes and it's a reversal membrane potential, it's an action potential. And then the muscle responds by contracting. And this is basically what you see in these, what we're going to call pacemaker cells, not the contractile cells, the pacemaker cells. The pacemaker cells are going to contract. They're going to spontaneously depolarize, right? So the contractile cells spontaneously depolarize, and then they repolarize, right? This is our potassium channel, and they go below threshold, and then instead of staying at rest, they start to creep. Notice towards threshold, and they creep, and then they hit threshold again, and then they produce an electrical current. It goes down. And then they creep, and they produce an electrical current. Each electrical current produces a contraction. So this is what happens in all cardiac muscle cells. They creep. They never rest, if you will. They never stay at resting membrane potential. They start creeping immediately. And that's because some of these channels that they have are going to close and open spontaneously. And we'll talk about how and why here in a minute. These are called pacemaker potentials. They call it funny current is the name that physiologists gave to it, because it's a current you don't see anywhere else, and these cells never rest. And so the channels, they even call them funny channels because these channels are weird. They open and close when they're not supposed to. Now you can change the basic rhythm of the heart by using chemical ligands or communication molecules. Acetylcholine and epinephrine, for example, are two of these chemicals. Caffeine's another one. In my experiment that we'll do in this lab that I'll send to you virtually, that I've done it down here in my basement, I'll show you how different chemicals affect heart rate and contraction, force of contraction. But acetylcholine and epinephrine are two major players in speeding up and slowing down the heart. They're antagonists to each other. Epinephrine, or adrenaline, speeds up heart rate. It's called the cardiac accelerating hormone, where acetylcholine slows down heart rate. Acetylcholine is released by the parasympathetic nervous system via the vagus nerve, and it slows heart rate. Epinephrine is released by cardiac accelerator nerves and is part of the sympathetic nervous system. And so these two can speed and slow down the heart. So if your heart's not contracting fast enough for your central nervous system's monitoring heart rate, for example, it can speed it up. If it's contracting too much, it can slow it down, right? But the heart, typically, if it's working just fine for whatever activity you're doing, like sitting and studying, your heart's controlling its own rhythm. The nervous system only kicks in or jumps in if we need a change in contraction and flow rate of blood. We'll talk about how acetylcholine slows down heart rate and how epinephrine speeds up heart rate at the end of this chapter. So let's talk about these funny currents, these cells, these pacemaker cells, and how they spontaneously depolarize. So these cells have specialized channels, and they are called HCN channels. All right. These channels are found in pacemaker cells, not contractile cells, in the pacemaker cells. They've also been discovered in some other places in the body. In particular, they've been discovered in the brain. They form some of the circuits in the brain and allow for spontaneously depolarizing of certain circuits for memory, short-term memory, long-term memory, and in some behaviors in the hippocampus and some other parts of the brain. But these HCN channels, what they're called, are hyperpolarizing nucleotide channels. Hyperpolarizing nucleotide channels. They're called nucleotide channels because they typically can bind CAMP and cyclic GMP also. Oops. So they can bind these nucleotides, which we call CAMP, which is cyclic AMP, right? Which is this cyclization of ATP is the major one. And what CAMP does is can desensitize them, they open slower, or hypersensitize them and they open faster. And so it just depends on which of these we're talking about and where they're located. Most pacemaker cells in our hearts, in the mammalian heart, have what are called HCN4 channels. There are a bunch of subtypes. And what these channels do, as you can see, is they spontaneously are going to depolarize here, as you can see in this animation. And we're talking about these HCN channels are the green channels. All right. And there's a lot of debate on what these channels are and which ones are which. But there are two major HCN or pacemaker or funny current channels. There are sodium channels, HCN sodium channels, and the HCN calcium channels. Now, I will tell you this. The potassium leakage channels, which are controlling resting membrane potential, in cardiac muscle cells, a few of them are gated. And they close at really negative membrane potentials. And so when these pacemaker cells completely repolarize and they hit that negative 60, this is where some of the potassium channels close. And if we close potassium leakage channels, then not as much potassium is leaking. And so the cell starts creeping to the positive. And when we start getting to about negative 55, then we open these HCN sodium channels. All right, these HCN sodium channels open and close. They open and close. And so they are going to cause basically about this much of this pacemaker potential. The first part here, this first creeping part in blue, is because of the closing of some of the potassium leakage channels. So some of these close because they're gated. So some of the potassium leakage channels close. When they get up to about negative 55 and close, that opens these HCN sodium channels. And then when you get up to about negative 45, then right here in red, that's when these, what are called T-type calcium channels open. All right, the T-type calcium channels are called transient calcium channels. They open and close. I mean, these HCN sodium channels and these T-type calcium channels, they open and close almost immediately. Once they open, they close. And so only a little calcium comes in, but it's enough to push at the threshold, right? Once you hit threshold, then you get what are called L-type calcium channels. So notice L-type, all right? So T-type calcium or the HCN calcium channels are different than the L-type calcium channels. L-type means long-lasting. And so these calcium channels, they open longer, right? Lots of calcium means L-type. And so you go from negative 40 up to positive 20. And then they close. And then potassium channels here are going to repolarize the cell back to negative. And then some of the calcium channels close. See that? Those are those little, the leakage channels close. Then the sodium channels open again and close, and then the calcium channels, T-type calcium channels open and close, and then bam, the L-type channels again. So you're always creeping. You're never maintaining rest, and that's because of these HCN channels. They're only found in the pacemaker cells, not the contractile cells. This is explaining the action potentials in the autorhythmic cells or the pacemaker cells only, not the contractile cells. So again, I need you to learn this, write it out, and understand that because we have HCN channels here, these cells behave differently. They produce their own spontaneous electricity. And so we have basically a conducting system here that is going to be made up of what are called autorhythmic cells. So these cells have the ability to spontaneously depolarize. And the fastest spontaneously depolarizing cells, these cells that have these funny currents that we talked about earlier, that can spontaneously reach threshold and cause an action potential, the fastest ones, the ones that do it the quickest, are going to be found in the wall of the right atrium in a area called the sinoatrial node. Sino meaning in the sinus side, or the wall of a cavity, the right atrium, and atrial meaning the atrium. These cells will spontaneously depolarize, and the atrial network of cardiomyocytes that contract, they're called contractile cells, actually attach to the SA node through gap junctions. There's also a set of autorhythmic cells that conduct the action potential directly to the next group of spontaneously depolarizing cells, which are called the AV node, the atrioventricular node, because it's found in the septum, or the wall between the atria and ventricles. There is no direct connection or gap junction connection between the contractile cells and the AV node. So the SA node, what it does is it spontaneously depolarizes the entire atrial network, and right after the SA node depolarizes, all of the atria, or both atria, contract, and they force blood through the valves, the atrioventricular valves. The one on the right side is called the tricuspid valve. The one on the left side is called the bicuspid valve. They're going to force those valves open and fill the ventricles with blood. Also, there are tracts called internodal tracts. If I can open my marker here. Internodal tracts that run from here and here to the AV valve. Those internodal tracts deliver the action potential from the SA node directly to the AV node. Those will cause the depolarization of the AV node, which then doesn't send the action potential directly down into the ventricles first. It actually sends the signal through another group of conducting cells that are called bundle branches, or they're called the bundles of His. Whoops. So the bundles of His, actually, after the scientists who discovered them, and these bundle branches, they divide, and one will feed the right side of the heart and one will feed the left side of the heart. As they come down through the septum in the middle between the right and left ventricles, they will divide into Purkinje fibers, which are very thin fibers that then innervate this muscle mass evenly. The problem with the ventricles are they're really thick and there's a lot more muscle mass to it than the atria. And so we want to not only spread the action potential evenly, but we want to start the action potential spreading to the muscle on the apex. So the apex contracts first and pushes upward. So the blood goes up and into the blood vessels that come out of the top of the heart. We don't want the blood to contract down first and then come up. That's why we spread the action potential to the base or apex of the heart, the bottom of the heart, and then we squeeze upward. So contraction starts here and works its way up, forcing blood up. The blood will come up, close the valves, and then be forced into the aorta on the left side and pulmonary circulation on the right side. So it's a little more complicated as far as the conducting system in the ventricles because we want to spread the signal evenly and we want to start contraction at the apex or at the pointing end of the caudal end of the heart. One other thing that's really interesting is there's a delay here in the AV node, right? And so that delay allows for the atria to completely contract before the ventricles are stimulated. So these are called pacemakers. The SA node in particular is the major pacemaker and controls heart rate. Typically, the SA node or the pacemaker depolarizes anywhere from 60 to 80 times per minute, and you get 60 to 80 contractions of the heart per minute. It's the fastest of the depolarizing pacemaker cells. So the heart is myogenic. It produces its own rhythm, its own contraction rate. All right, so this is a blow up right here of the SA node. So we blow this area of the heart of the right atrium up. And so here are the cells, the autorhythmic cells of the SA node. They start the electrical current with their funny current, right? And their generated action potentials at about 60 to 80 times per minute. And that will spread to the atrial network, right? Through gap junctions and depolarize both atrial networks at the same time, which will cause even and coordinated contraction and pushing of blood into the ventricles. There are also internodal pathways that leave the SA node and deliver the action potential directly to the AV node so that we can stimulate the ventricles. The AV node has no connection other than those internodal pathways to the SA node. And so in order to coordinate the atrium ventricles, we need those internodal pathways, or the AV node will cause contraction at a different rate than what we need.